is one of the techniques to be used for future revival of extinct species. De-extinction, also known as species revivalism or resurrection biology, is the process of regenerating extinct species. The idea of de-extinction has been there for some time and has been received worldwide attention through science fiction movies such as Jurassic Park. Bringing back such animals is both thrilling and terrifying. In real life, de-extinction is, however, incredibly challenging, and it remains uncertain whether dinosaur DNA is recoverable. Current technology can only utilize DNA samples of 1 million years old. Theoretically, scientists can bring back a Ne- that Neanderthal, Neanderthal, but not a Triceratops, as they last roamed the surface of the earth 65 million years ago. Approaches to de-extinction include cloning, backbreeding, and genome editing. Species that have been considered for de-extinction are the woolly mammoth, Pyrenean ibex, roach, thylacine, quagga, and the passage pigeon. Cloning. Cloning is a term used to refer to somatic cell nuclear transfer, a technique that is used to create a genetic copy of an organism. It is among the most common methods suggested for the restoration of extinct species. The somatic cell near transfer is a multi-stage procedure. Somatic cells are first harvested and cultured in vitro before nuclei are extracted from the cultural cell. Egg cells are concurrently harvested from a species that is closely related to the target animal. The enunciated, the enucleated, the enucleated egg is then fused with the extracted nucleus from the somatic cell. After cell division, the embryo is then implanted into a surrogate maternal host, which later gives birth to the genetic copy of the animal. Dolly, a sheep born in the mid 1990, is perhaps the most well known example of a cloned animal. Cloning is an attractive de-extinction approach as it results in an organism that is identical at the nuclear genome level to the donor of the somatic cell. Intact living cells are, however, required for cloning. Unfortunately, most extinct species do not have intact living cells. That limitation means that only recently extinct species can undergo de-extinction through cloning from cultural and frozen cells that have been collected before death. Cloning of extinct species has been tried before with considerable success. In 2003, a cloned bucardo, an extinct subspecies of the Ibex was born to a surrogate mother 9 years after the death of the last individual. Years earlier, French, Spanish, and Belgian scientists had managed to harvest a skin sample of the last members of the subspecies, a female called Celia, from which cells were cultured and then frozen. A majority of the reconstructed embryos failed to develop to term. Out of this process, a single bucardo was born but unfortunately died after birth due to a lung deformity. Backbreeding. Backbreeding is the use of selective breeding to enhance certain ancestral traits in existing population of living organism. The technique aims to concentrate the desired ancestral traits that persist in a population into one individual. Mating pairs are selected if they display a desired behavioral or morphological quality. Gradual backbreeding eventually leads to the resurrection of traits that had previously been lost and diluted over time. The technique, however, has several limitations as a de-extinction approach. For example, the method requires that target ancestral traits persist in living species. Backbreeding can also lead to high levels of inbreeding or the creation of undesired combination of alleles, which can lead to an overall decline in the population fitness. Backbreeding in the past has been used to try and bring back a roach, an extinct species that is related to modern day domestic cattle. Experts believe that the species had forward facing horns and aggressive temperament and was the larger than the present day cow. The traits can be found today dispersed across various breeds of cattle. The first attempt of this de-extinction of roach began in the 1920s. Lutz and Heinrich Heck, both directors of a German Jew, started the back breeding project that led to the creation of a Heck cattle. The cattle had a primitive appearance. The cattle had a primitive appearance and were more aggressive. They were, however, not considered successfully bred. Experts note that they lacked the roach distinctive morphological traits. Today, scientists are trying to bring back roach using genetics to guide the process to ensure a higher success rate. Genetic engineering. Genetic engineering takes advantage of advances made in the field of genome editing and DNA technologies. Advances in DNA sequencing and ancient DNA extraction technology are increasingly making it feasible to construct complete genome sequences of extinct species. With the help of such technology, scientists can align genomes to the genome sequences of closely related living species. Differences between the living relative and extinct animal genome are then edited through genome engineering, resulting in living cells with the extinct genes. The living cells can then be used for cloning in species that have been extinct over a long time. The reconstruction of the genome sequence is recoverable from various tissue types since DNA survives despite increasingly complex environments. This technique to be 7 lakh years old. The technique has made the resurrection of genome sequence sequences belonging to samples of various extinct species possible, including mammoths. The method, however, has several limitations, including difficulty in recovering DNA from remains found in hot and wet environments. Genome from extinct species and those lacking close living relatives may also be challenging to assemble. How strict is the extinction? It is important to note that none of the approaches listed lead to the selection, resurrection of an organism that is precisely identical to the extinct species. For example, organisms that are cloned from frozen cells collected from extinct species share a nucleus genome but also have mitochondria from the nucleated eggs, which belongs to a different animal. Genome editing leads to similar genes responsible for a phenotype, but the genes are expressed as part of different genome expressed as part of the different genomic background. Precise replication of extinct species is, however, not necessary when de-extinction is aimed at conservation. The majority of de-extinction procedures currently being carried out are aimed at creating functional equivalent of the extinct species. Ecological proxies capable of occupying the ecological niche left by the extinct species. Ethics. Several ethics of de-extinction have raised ethical issues as a reason as to why such projects should be discouraged. Motives of de-extinction have been questioned. Are we bringing back extinct species because we feel guilty for driving some of them to extinction? Are we seeking restoration justice? Is that case? Who is the justice for? Critics believe that justice is not for the individuals of species that have undergone de-extinction because they might suffer from malformation and maladjustment. Atoning for our ancestors' action by potentially causing further suffering to resurrected individual animals therefore poses an ethical dilemma. Ethology. Various animals, including mammals and birds, learn behavior by watching and interacting with other members of the species. De-extinction presents the challenge of having organisms that do not have other old individuals of the same species to interact with and learn from. Captive condor breeding programs have in the past demonstrated the dangers of not having parents from the same species. Despise humans using puppets or as parents to ensure that the words do not imprint on humans. The captive birds, once released into the wild, displayed an unhealthy curiosity for humans. The birds were also less social with other members of the species. Ecology. The reintroduction of extinct species that have undergone de-extinction into the wild could also be unintended consequences on the overall ecology as we know it. For example, similar attempts on living species, such as the gray wolf reintroduction into Yellowstone National Park, led to wide ranging ripple effects, including the reduction of deer and elk population, which led to the of aspens. Thriving of aspens. Econom- Economics. Experts believe that apart from the cost related to de-extinction, maintaining individuals that have been brought back could also be a very costly affair. For example, maintenance costs associated with a healthy elephant are $70,000 per year. Maintenance of animals gets more expensive as they get older. One must therefore consider long-term maintenance cost of the individual that have undergone de-extinction. Otherwise, we risk having a situation whereby individuals are euthanized once the excitement wears off and funding runs low. De-extinction, a method of conservation. According to Professor Eugene or Cindy Tian of the University of Connecticut, with sufficient funding and political will, it might take only a decade to have use with rare and endangered animals. In 2000, planned to display a clone Gore named Noah at the San Diego Zoo were not realized after the animal got an infection and died after 2 days. The U, however, managed to house a cloned Benteng for 7 years before it was euthanized. Advocates believe that de-extinction is the next step in the conservation. Extinct species can be can be brought back to fill ecological function that have been left vacant. Experts believe that organizations such as the IUCN are best suited to regulate de-extinction in the future, also called resurrection biology. De-extraction, the process of resurrection species that have died out or gone extinct, although once considered a financial notion, the possibility of bringing extinct species back to life has been raised by advances in selective breeding genetics and reproductive cloning technologies. Key among those advances was the development in the 1990s of a technique known as somatic cell nuclear transfer, which was used to produce the first mammalian clone, Dolly, the sheep born 1996 and died 2003. In 2009, using SCNT, scientists very nearly achieved de-extinction for the first time, attempting to bring back the extinct Pyrenean ibex. A clone was produced from preserved tissue, but it died from a severe lung defect within minutes of being born. The near success of the attempt sparked debate about whether species should be brought back from extinction, and if they are brought back, how it should be done and how the species should be managed. The candidates species of species for de-extinction are many. Some high-profile examples include the woolly mammoth, the passenger pigeon, the thylacine, and the gastric brooding frog. De-extinction does not extend to dinosaurs, partly because of the extreme old age of specimen and the severe degradation of DNA over time. Tools of the tools of species resurrection. The tools of species resurrection. The possibility of bringing extinct species back to life was first explored in the early 20th century through an approach known as backbreeding. Backbreeding for the production of a breed that displays the traits of a wild ancestor is based on the principles of selective breeding, which humans have used for centuries to develop animals with desired traits. In the 1920s and 30s, German geologist Lutz and Ines Heck crossbred different types of cattle in an attempt to backbreed for an animal that resembled the roach, an extinct species of European wild ox ancestral to modern cattle. The Heck brothers crossbred modern cattle based on historical description and bone specimen that provided morphological information about the roach but had no insight into the animal genetic related, relatedness. As a consequence, the resulting Heck cattle bore little resemblance to the roach. In the later part of the 20th century, tools emerged that enable scientists to isolate and analyze DNA from the bones, hair, and other tissues of the dead animals. Coupled with advances in reproductive technologies such as in vitro fertilization, researchers were able to identify cattle that are close genetic relatives of the roach and combine their sperm and eggs to produce an animal that is morphologically and genetically similar to the roach. Other advances in genetic technology have raised the possibility of inferring and reconstructing the genetic resource sequences, the genetic sequences of extinct species from even poorly preserved and cryopreserved specimens. Reconstructed sequences could be compared against the sequences of extant species, allowing for the identification of not only living species or breeds best suited for backbreeding, but also genes that would be candidates for editing in living species. Genome editing, a technique of synthetic biology, involves adding or removing specific species of DNA in a species genome. The discovery of CRISPR, a naturally occurring enzyme system that edits DNA in certain microorganisms, greatly facilitated the refinement of genome editing for de-extinction. Cloning of de-extinction has centered on the use of SCNT. SCNT entails the transfer of the nucleus from a somatic cell of the animal to be cloned into the cytoplasm of an nucleated donor egg, an egg cell that came from another animal that has had its own nucleus removed. The egg cell is stimulated in the laboratory to initiate cell division, leading to the formation of an embryo. The embryo is then transplanted into the uterus of a surrogate mother, which in the case of the extinction is a species closely related to the one that is being cloned. The attempt to resurrect the extinct Pyrenean ibex in 2009, researchers transferred nuclei from third fibroblast of cryopreserved skin specimens into nucleated eggs of domestic goats. The reconstructed embryos were transplanted into either Spanish ibex or hybrid females. It may be possible to use stem cells to resurrect extinct species. Somatic cells can be reprogrammed through the introduction of specific genes, creating so-called induced pluripotent stem. Pluro, pluro, Pluripotent stem (iPS) cells. Such cells can be stimulated to differentiate into different cell types, including sperm and eggs, that potentially can give rise to living organisms. Similar to the other techniques of de-extinction, however, the success of an approach based on stem cell depends largely on the quality of DNA that is available in preserved specimen. Ethical consideration: cloning, stem cell manipulation, genome reconstruction, and genome editing are powerful technology with significant ethical ramification. When applied to de-extinction, the expenses and inefficiency of SCNT, for example, has raised questions about its practicality for resurrecting extinct species. Perhaps the greatest concern Perhaps the greatest concern, however, is the potential of those technology as well as backbreeding to alter the course of natural history. De-extinction provides an opportunity for humans to rectify post harms inflicted on other species as well as the expand species diversity. But many extinct species were driven out of the existence as a result of habitat loss and other lived in habitats that have since been altered dramatically. In addition, in the near term, resurrected species would be considered endangered and would therefore require conservation, for which resources often are constrained or lacking. De-extinction, by providing the option to bring species back later, also could have the unintended consequences of condoning extinction and could give impetus to endeavors that threaten biodiversity. Other concerns include unknowns about the fate of resurrected animals, from the health of cloned individuals to whether the animals would be able to adapt to current environmental conditions and whether they would be able to produce viable offspring. The classification of species revived through backbreeding, cloning, or genetic reconstruction, all of which could involve divergence from an extinct species' original genetic constitution, also remains uncertain. The potential to be leveraged as a means of advancing financial and commercial interest has led some to question the motivation of researchers and companies behind certain de-extinction projects. Nonetheless, de-extinction has helped fuel important progress in science, building particularly on knowledge in developmental biology and genetics. It also has generated interest in endangered species, with many of the tools of de-extinction also being applicable to conservation of endangered species. The construction of extinct genes could be used, for example, to restore genetic diversity in threatened species or subspecies. Thank you. Please subscribe, like, comment, and share.